Friends, we are discussing a new chapter today, right? It is biotechnology principles and processes, right? Go for introduction. Technology means application of science. What is the meaning of technology? Application of science. Sir, bio, you know it, living. Therefore, technology means application of science. Bio means living. Therefore, application of science to living organisms. This is simple definition for biotechnology. Is it okay? Right? Now listen here. Biotechnology is defined as utilizing living organisms. What? Utilizing living organisms. Utilizing what, sir? Living organisms or enzymes of living organisms. What? Enzymes. Enzymes of living organisms. Why? To produce a product. To produce a product which is useful to human. Which is what, sir? Which is useful to human. See this definition, sir. What we are using? We are utilizing living organisms or enzymes of living organisms. Why we are utilizing living organisms or enzymes of living organisms? To produce a product. Which product, sir? The product which is useful to human beings. If you proceed according to this definition, then bread making, curd preparation, wine preparation, all this comes under biotechnology, because all these processes, they involves microorganisms in them. According to this definition, if living organisms are utilized, we should call it as what? We should call it as biotechnology. If living organisms are utilized to produce a product which is useful to human, sir, microorganisms are involved in the production of bread, curd, wine. Yes or no? Sir, this bread, curd, wine, they are useful to human beings. Therefore, we are producing products which are useful to human beings by utilizing living organisms. So, this bread preparation, curd preparation, wine preparation, they comes under biotechnology if we proceed according to this definition. Is it okay? Clear? Right. Therefore, according to this definition, is it okay? Right. Therefore, in this sense, all microbial mediated process which are carried out with the help of microorganisms, such as what? Such as bread, curd and wine preparation comes under biotechnology. Comes under what, sir? Biotechnology. But strictly speaking, bread preparation, curd preparation, we cannot call it as biotechnology. Even though what are involved? Microorganisms are involved. Even though they are useful to human beings. That's why at present, it means nowadays, the word biotechnology is used in restricted sense. The term biotechnology is used in restricted sense. Is it okay? Right? Therefore, today, the term biotechnology is used in a restricted sense. The term biotechnology is used in a restricted sense. Is it okay? Right? Sir, we are saying that we are using the term biotechnology in a restricted sense. What it is, sir? Take a genetically modified organism. If that genetically modified organism produces a product which is useful to human beings on large scale, then we are going to call that process as biotechnology. Which process now we are calling as biotechnology? That process should involve genetically modified organisms. By using that genetically modified organisms, we have to produce a product. Which product? Which is useful to human beings on large scale. Is this statement okay? Right. Therefore, that is using what? Genetically modified organisms. Using what, sir? Genetically modified organisms. Using genetically modified organisms. Why? To produce a product. To produce a product which is useful to human. To produce a product which is useful to human on industrial scale. What is the meaning of industrial scale? Large scale. Large amounts. Therefore, using what, sir? Genetically modified organisms. Why? To produce a product which is useful to human on industrial scale. At present, we are calling such process as biotechnology. Which process? The process which uses genetically modified organisms to produce a product which is useful to human on industrial scale. Is these statements okay? Is it okay? Right? Now listen here. If we proceed according to this definition, then bread preparation, curd preparation, wine preparation won't come under biotechnology. Do they come under biotechnology? No. Therefore, according to this definition, right? Now listen here. Therefore, according to this definition, that is in this sense, right, according to this definition, right, what preparation? Bread, curd and wine preparation does not comes under biotechnology. Do they come under biotechnology according to this definition? No, because curd preparation, right, bread preparation and wine preparation, they involves microorganisms, but that microorganisms are not genetically modified. Are they genetically modified? No, they are not genetically modified. Is it okay? Therefore, they won't come under what, sir? They won't come under biotechnology. What? Bread preparation, curd and wine preparation. Then what comes under biotechnology? According to this definition, biotechnology includes number one, in vitro fertilization. What, sir? In vitro fertilization. Sir, what is the meaning of fertilization? Fusion of male gamete with female gamete. What is fused with what? male gamete is fused with female gamete. What is that process called as? Fertilization. Sir, in vitro, artificial conditions, laboratory conditions, aseptic conditions, yes or no? It means male gamete is fused with female gamete, right, in vitro conditions. Okay, that is in laboratory conditions inside the test tube. Sir, if male gamete is fused with female gamete, what is formed? Zygote. Zygote develops into embryo. Embryo develops into a new offspring. Yes or no? Therefore, in vitro fertilization, which leads to test tube baby, which leads to what, sir? Test tube baby, which leads to test tube baby. What? In vitro fertilization, fusion of male gamete with female gamete inside the test tube, right, inside the test tube in vitro conditions, forms a zygote in the test tube. Zygote develops into embryo, right? This embryo develops into a new offspring. We are calling it as what? Test tube baby. Is this statement okay? Right. Therefore, what is the first one? In vitro fertilization, which leads to, right, test tube baby. In first year, we read one statement. Panchanan Maheshwari, what is the scientist's name? Panchanan Maheshwari is associated with in vitro fertilization of angiospermic plants. Which plants? Angiospermic plants. I am not speaking about test tube baby. Is it okay? I am not speaking about animals. I am speaking about angiospermic plants. Panchanan Maheshwari is associated with in vitro fertilization of angiospermic plants. Is it okay? Right. Next one, number 2, artificial synthesis of a gene. Artificial synthesis of a gene. It means to synthesize the gene artificially and using that gene. Therefore, synthesizing the gene artificially and using that artificially synthesized gene, that comes under biotechnology. Is it okay? Sir, who is the first scientist to synthesize a gene artificially? Harigovind Khurana. What is the scientist's name? Harigovind Khurana. He is an Indian born American scientist who synthesized a gene artificially for the first time. Therefore, Harigovind Khurana is an Indian born American scientist, right, who first synthesized the gene artificially. Is it okay? Right. Next, number 3. What it is? Developing DNA vaccines. Developing what, sir? DNA vaccines. Developing DNA vaccines. And next, number 4, correcting defective gene. Correcting what? Defective gene. Correcting which gene, sir? Defective gene. Now listen this statement. Sir, if any defectiveness is there in a gene, that defective gene is replaced with a correct gene. Therefore, non-functional gene is replaced by a functional gene. Defective gene is replaced by a effective gene. This therapy is called as gene therapy. What is this therapy called? As a gene therapy. It is called as gene therapy. I already told you, at present we are using the term biotechnology in a restricted sense. What is that, sir? Using genetically modified organisms. Using genetically modified organisms to produce a product on industrial scale, on large scale, and that product should be useful to human beings. Is it okay? According to this definition, this comes under biotechnology, but bread preparation, curd preparation and wine preparation, they won't come under biotechnology. They won't come under biotechnology. Is it clear? Right? Here. Old biotechnology. What? Old biotechnology is based on capacity of microorganisms. Which biotechnology? Old biotechnology is based on what, sir? Capacity or capacities of microorganisms. Sir, new biotechnology. What, sir? New biotechnology. New biotechnology is based on genetic engineering. New biotechnology is based on what, sir? Genetic engineering. Genetic engineering. Sir, what is another name for genetic engineering, sir? I already told you it is also called as a recombinant DNA technology. What is another name for, right, genetic engineering, sir? Recombinant DNA technology. That is rDNA technology. Recombinant DNA technology. Nothing but rDNA technology. Therefore, old biotechnology is based on capacity or capacities of microorganisms, whereas a new biotechnology is based on what, sir? Genetic engineering. A organization by name European Federation of Biotechnology. European Federation of Biotechnology. This statement is important. European Federation of what? Biotechnology. In shortcut, this organization is written as EFB. What is the full form of EFB? European Federation of Biotechnology. Defined biotechnology. Defined biotechnology as what, sir? Utilizing what? Utilizing properties. Utilizing properties and uses of microorganisms. Utilizing properties and uses of microorganisms are to exploit cell and cell constituents. Cell and cell constituents at industrial level. At industrial level to produce a product which is what? Which is essential to life or useful to human beings. This is the definition given by European Federation of Biotechnology. What it is? Utilizing properties and uses of microorganisms. Utilizing what, sir? Properties and uses of microorganisms, or to exploit cell and cell constituents. To exploit cell and cell constituents. Why? Right? To produce a product which is essential to life or which is useful to human on industrial level. Industrial scale means large quantities in large amounts. Is this statement okay? This is a definition given by European Federation of Biotechnology. Is this statement okay? Right? European Federation of Biotechnology, that is EFB, right? Defined biotechnology. Defined biotechnology. How, sir? By combining by combining a traditional view by combining traditional views with modern molecular emphasis. Is this statement okay? Therefore, European Federation of Biotechnology defined biotechnology how, sir? By combining the traditional views with a modern molecular emphasis. Modern molecular emphasis. The two core techniques. The two core techniques which gave birth to biotechnology. The two core techniques which gave birth to biotechnology are, number one, first one, genetic engineering. What's that? Genetic engineering. Number two, tissue culture. Tissue culture. Therefore, the two core techniques which gave birth to what's our modern biotechnology, which gave birth to modern biotechnology are genetic engineering and what is the second one, sir? Tissue culture. Genetic engineering and what? Tissue culture. Is these statements okay, right? We'll go for the next topic, construction. Construction of a first artificial rDNA, that is a recombinant DNA molecule. Is it okay, right? In 1972, in which year? 1972. Cohen and Boyer. What are the two scientists' names? Cohen and Boyer. In 1972, Cohen and Boyer, they constructed a first artificial rDNA molecule. Therefore, who was the first to construct a artificial rDNA molecule? Cohen and Boyer. In which year, sir? 1972. How they constructed this rDNA molecule, sir? Before going to that, first you should know about a word, plasmid. In first year, you read about this word plasmid, yes or no? Right? Sir, plasmid, how we are going to define this plasmid, sir? Plasmid is extra chromosomal, right? We got it in first year. Extra chromosomal, circular, naked, self-replicating, double-stranded DNA molecule, right? Double-stranded DNA molecule present in almost all bacterial species. Present in almost all what? Almost all bacterial species. Is this statement okay? Right. Sir, plasmid is extra chromosomal, circular, naked, self-replicating, double-stranded DNA molecule present in almost all bacterial cells. Is this word okay? What it is? Plasmid. Therefore, sir, plasmid is present in almost all bacterial species. Yes or no? Right. Next important statement. Sir, plasmid contains antibiotic resistant genes. I am not saying that a plasmid contains antibiotic producing genes. Plasmid contains antibiotic resistant genes. It means plasmid contains which genes? Antibiotic resistant genes. Is it okay if that antibiotic resistant genes are present in a plasmid, automatically the bacteria gets resistance against antibiotics, Is it okay? Right. Now we are seeing the construction of first artificial rDNA molecule, right? Now see here, what Cohen and Boyer they did means, from a bacteria they isolated the plasmid. From where, sir? Bacteria. What they did? Isolated. What is isolated? Plasmid. Sir, plasmid means double-stranded DNA molecule, right? Sir, DNA, what is the segment of DNA called as genes? One of the gene present in this double-stranded DNA of plasmid, right, is antibiotic resistant genes. I already told you plasmid contains antibiotic resistant genes, yes or no, right? Sir, from bacteria, Cohen and Boyer, they isolated the plasmid. Plasmid means a double-stranded DNA. From this plasmid, right, they isolated the what's our antibiotic resistant gene. What is isolated, sir? Antibiotic resistant gene. Isolated. Antibiotic resistant gene was isolated. Now see this statement, sir, plasmid having number of genes. From these genes, what they isolated? Antibiotic resistant gene. How, sir? By cutting the plasmid. By cutting the plasmid. Sir, how they made a cut in the plasmid, sir? By using restriction endonucleases enzymes. Which enzyme, sir? Restriction endonucleases enzymes. By using restriction endonucleases enzymes, they made a cut in the plasmid. Once they made a cut in the plasmid, right, they can isolate an antibiotic resistant gene. Therefore, is these statements okay, clear? Right. What is the enzyme name, sir? Restriction endonucleases, right? Restriction endonucleases are also called as molecular scissors. Restriction endonucleases. Restriction endonucleases are also called as a what's a molecular scissors. Also called as molecular scissors. They are also called as a chemical. Also called as chemical knives. Which knives, sir? Chemical knives. They are also called as a chemical scalpels. Also called as chemical scalpels. Is it okay? Therefore, restriction endonucleases are also called as molecular scissors or chemical knives or chemical scalpels. Is this statement okay? Right. Sir, Cohen and Boyer, what they did? Isolated a plasmid from a bacteria. Is it okay? Right. From this isolated plasmid, they isolated the antibiotic resistant gene by using restriction endonuclease enzymes. Is it okay? Right. Now this antibiotic resistant gene that was isolated from the plasmid, this is the desired gene. In this case, what is the desired gene, sir? Antibiotic resistant gene. From where it is isolated? From plasmid. From where that plasmid was isolated? It was isolated from a bacteria, right? Now Cohen and Boyer, what they did means, they took another bacteria by name Salmonella. What it is? Salmonella typhimurium. What is the bacteria name? Salmonella typhimurium bacteria. Salmonella typhimurium bacteria. Is it okay? From the Salmonella typhimurium bacteria, right, Cohen and Boyer, they isolated a plasmid. What they isolated? Plasmid. Sir, plasmid means linear or circular? Plasmid means it's circular, right? Therefore, isolated which plasmid, sir? Circular plasmid. Therefore, isolated, what sir? Isolated, what they isolated? Plasmid. Isolated plasmid. Sir, how this plasmid is, sir? Circular, right? Circular. Sir, this circular plasmid was made linear. What was made linear? Circular plasmid was made linear. Sir, how a circular plasmid can be made linear, sir? By cutting the circular plasmid by using restriction endonucleases. Therefore, by using restriction endonucleases, molecular scissors, okay, if you make a cut in the circular plasmid, then circular plasmid becomes linear. Now this is a linear plasmid. This is which plasmid? Linear plasmid. If two ends are not joined, then we can call it as a linear. Linear always won't mean that it should be like this. Is this statement okay? If two ends are not joined, we can call it as what, sir? Linear. Therefore, generally plasmid is circular like this. When we used restriction endonucleases, it has made a cut in the circular plasmid. Once it makes a cut, right, then two ends, are they joined? No. So now this is which plasmid, sir? Linear plasmid. Therefore, circular plasmid has become what? Linear plasmid. Linear plasmid. How circular became linear, sir? By using restriction endonuclease enzymes. By using which enzymes? Restriction endonuclease enzymes. Now, is this statement clear? Okay, right. Now you tell me, in this experiment, right, who is acting as a desired gene? Antibiotic resistant gene. Is it okay? Right. In this process, this plasmid acts as a vector. Who acts as vector? Plasmid. This plasmid acts as vector. Vector means carrier. Vector means what? Carrier. Now listen this statement. Sir, this is circular plasmid. By using restriction endonuclease enzymes, they made a cut. Now it is linear or circular, sir? Now it is linear. Sir, now what Cohen and Boyer did means, this is antibiotic resistant gene. They inserted this antibiotic resistant gene right in this linear plasmid. Is this statement okay? Therefore, into the plasmid, what they inserted, sir? They inserted antibiotic resistant gene. Now you tell me, sir, how this end of the plasmid is joined with this ends right of the antibiotic resistant gene, sir? See this statement carefully. Sir, this is one end of what, sir? Plasmid. This is one end of what, sir? Antibiotic resistant gene. These two ends should be joined, yes or no? Similarly, this is the end of the plasmid. This is the end of the antibiotic resistant gene. These two ends should be joined, yes or no? Right. This joining takes place with the help of one enzyme by name DNA ligase. What is the enzyme name? DNA ligase. Is this statement clear? Is it okay? Right. Therefore, Cohen and Boyer, what they did means, they inserted this antibiotic resistant gene into this linear plasmid. Into the linear plasmid. Into what, sir? Linear plasmid. Then what we got? We got rDNA. What we got, sir? We got rDNA. Sir, this joining antibiotic resistant gene with linear plasmid, it is called as a ligation. Ligation. This ligation is done with the help of which enzyme, sir? DNA ligase. DNA ligase is also called as a molecular glue. Who is molecular scissor restriction endonuclease? Who is called as a molecular glue DNA ligase? What is the meaning of ligation? Joining. What is joined with what? Antibiotic resistant gene is joined with the linear plasmid. Antibiotic resistant gene is joined with the linear plasmid. Then what we got? rDNA. What is the full form of rDNA? Recombinant DNA. What it is, sir? Recombinant DNA. Recombinant DNA. Is this statement okay? This is the first artificially constructed rDNA molecule by whom, sir? By Cohen and Boyer. By whom, sir? Cohen and Boyer. Is this statement okay? Clear, right? Therefore, sir, this is which molecule, sir? Recombinant DNA molecule. Now you tell me, sir, recombinant DNA molecule, what it contains, sir? It contains a desired gene. It contains a vector. Yes or no? What it contains? Sir, it is having antibiotic resistant gene which is placed inside the plasmid. Here plasmid acts as what, sir? Vector. Therefore rDNA, what it contains? It contains a vector in which what is placed? Desired gene is placed. Therefore vector plus desired gene. These two together forms what, sir? rDNA, that is a recombinant DNA molecule. Therefore, vector plus desired gene is equals to rDNA. Vector plus desired gene is equals to rDNA. Is there any doubts in these statements, right? Now, listen here. Cohen and Boyer, now what they did means, they introduced this rDNA into the E. coli bacteria. Therefore, rDNA introduced into what, sir? E. coli bacteria. It means this E. coli bacteria acts as a host here. E. coli bacteria acts as what, sir? Host. Now listen these statements. Now where is rDNA, sir? Now rDNA is inside the E. coli bacteria. What is the meaning of rDNA? Vector plus desired gene. Vector plus desired gene. Yes or no? Now where is the rDNA, sir? It is inside the E. coli bacteria. It's inside the E. coli bacteria. Yes or no? Now see here. For example, this is a E. coli bacteria. What it is, sir? E. coli bacteria. It's a E. coli bacteria. Now see here, sir, this is which bacteria, sir? E. coli bacteria. It acts as host here. Then what is this one, sir? This is a rDNA, that is a recombinant DNA molecule. Now this rDNA present inside the E. coli bacteria replicates. What replicates? rDNA replicates. Where? Inside the E. coli bacteria. But replication requires one enzyme by name DNA polymerase. What is the enzyme name? DNA polymerase. Therefore, now rDNA replicates inside the host E. coli bacteria by utilizing the DNA polymerase of E. coli bacteria. Is this statement okay? Sir, rDNA, where it is replicating? Inside E. coli bacteria. But replication of DNA requires what? DNA polymerase enzyme. From where this enzyme is coming from? This enzyme is present in E. coli bacteria. Is this statement okay? Right? Therefore, inside what, sir? Inside right, E. coli bacteria. Inside E. coli bacteria. Inside E. coli bacteria. Right? What happens? rDNA replicates. rDNA replicates by utilizing DNA polymerase. By utilizing which enzyme, sir? DNA polymerase enzyme. By utilizing DNA polymerase enzyme present present in E. coli bacteria. Therefore, by utilizing DNA polymerase enzyme of E. coli bacteria, rDNA has replicated inside the E. coli bacteria. Now you tell me, if replication takes place, number doubles. If right, one rDNA is present, then it becomes 2, 2 becomes 4, 4 becomes what's sir 8. Therefore, number is doubled. See this statement carefully, listen it carefully, listen the statement carefully. Sir, rDNA means what plus what, sir? Vector plus desired gene, yes or no? If rDNA replicates, along with rDNA, what is replicating, sir? Desired gene is replicating. What is a desired gene here? Antibiotic resistant gene is a desired gene. It means here the desired gene has cloned or not cloned. The desired gene has cloned. Therefore, this cloning is called as what's our gene cloning. Therefore, what was the first gene that was cloned? Antibiotic resistant gene. What was the first gene that was cloned? Antibiotic resistant gene. Is these statements okay? Clear? Right. Therefore, inside E. coli bacteria, right, the rDNA has replicated, right, along with what? rDNA. Along with rDNA, desired gene replicates. Along with rDNA, what has replicated? The desired gene has replicated. Therefore, what is cloned? Antibiotic resistant gene was cloned. Therefore, what is the first gene that was cloned, sir? Antibiotic resistant gene. What it is? Antibiotic resistant gene. Antibiotic resistant gene, right? Basic steps that are involved in genetically modifying of an organism are 3 basic steps. 3 basic steps that are involved. 3 basic steps that are involved in genetically modifying of an organism. Are number 1, first one, identifying, right, what's that? Identifying DNA with the desired gene. Identifying the DNA with the desired gene. Therefore, identification of DNA with desired gene. Sir, in constructing the first artificial rDNA molecule, what was the desired gene? Antibiotic resistant gene. What was the desired gene? Antibiotic resistant gene. It was identified that antibiotic resistant gene was in the plasmid. Plasmid means nothing but a double-stranded DNA. Therefore, what is the first step? Identification of DNA with the desired gene. Is it okay? Right. Then what Cohen and Boyer did, sir? They placed this DNA, it means they placed the desired gene in a vector. After placing it inside the vector, then they introduced that rDNA into the host cell. Yes or no? Introduced rDNA into the host cell. We are discussing basic steps, not all the steps. Therefore, number 2, introducing DNA with desired gene introducing DNA with what, sir? Desired gene. Introducing DNA with desired gene. Where, sir? Into the host. Into the host. Actually, we are not introducing the complete DNA. Just we are cutting the DNA. We isolated the desired gene. That desired gene is placed inside the vector and rDNA is formed. That rDNA is introduced into the host. Is it okay? Right. Next, number 3, maintaining introduced DNA where it was introduced in the host. Therefore, maintaining introduced DNA in the host and transfer of this DNA to its progeny. Transfer of this DNA to its progeny. These are the three basic steps that are involved in genetically modifying an organism. These are the basic steps that are involved in genetically modifying an organism. Is it okay? Right. Now, main steps involved in genetic engineering process are: main steps involved in genetic engineering. Genetic engineering means nothing but what? rDNA technology, right? Genetic engineering, nothing but rDNA technology. Therefore, main steps involved in genetic engineering process are: number one, first one, isolation of a desired gene. Isolation of which gene, sir? Desired gene. Number two, insertion. Insertion of a desired gene into a vector. Insertion of desired gene into a vector. Next, number three, introduction of rDNA into host. Introduction of rDNA into host. Number four, selection of transformed cells. Each and every step we will discuss in detail, right? Just I am writing the side headings. Selection of what's are transformed. Selection of transformed cells. Next, number five, obtaining a foreign gene product. Obtaining a foreign gene product. Therefore, these are the main steps that are involved in genetic engineering. There is rDNA technology. Number one, isolation of a desired gene. Number two, insertion of desired gene into a vector. Number three, introduction of rDNA into the host cell. Number four, selection of transformed cells. Number five, obtaining a foreign gene product. Right? Go for the first step. What it is? Isolation of a desired gene. Isolation of a desired gene. Isolation of a desired gene. Right? Now listen here. For example, this is a desired gene, sir. Gene means what? Segment of DNA. Segment of DNA. Therefore, this is DNA, sir. Right? This is what? DNA. This DNA is with many segments. One of that segment is the desired gene. Is it okay? Right? Sir, DNA means it is in the chromosomes. DNA means where it is inside the chromosome. It is inside the chromosome. Sir, chromosome means where it will be inside the nucleus. Chromosome means it will be inside the nucleus. Sir, nucleus means it will be in the cytoplasm. Yes or no? It will be inside the cytoplasm. It will be inside what, sir? Cytoplasm. Sir, cytoplasm is surrounded by what, sir? Cytoplasm is surrounded by cell membrane or plasma membrane. I am saying cell membrane, not cell wall. This is cell membrane. What it is, sir? This is a cell membrane. This is cell membrane or plasma membrane. Sir, this cell membrane or plasma membrane is surrounded by what, sir? Cell wall. It is surrounded by cell wall. Is these statements okay? Cell wall. Is it okay? Sir, this is a desired gene. Gene means segment of DNA. DNA is inside the chromosomes. Chromosome is inside the nucleus. Nucleus is present inside the cytoplasm. Cytoplasm is bounded by cell membrane. Cytoplasm is bounded by cell membrane or plasma membrane. This cell membrane is covered by cell wall. I am speaking about plant cell. Okay. Therefore, cell membrane is covered by what, sir? Cell wall. Now see these statements carefully. If you want to isolate the desired gene, you have to remove all these barriers. To isolate what's our desired gene. What is the first step? Isolation of a desired gene. To isolate a desired gene, we have to remove all these barriers. Then only we can isolate the desired gene. What is the first barrier that is coming, sir? Cell wall. Therefore, first what we have to remove? We have to remove the cell wall. How we can remove the cell wall, sir? How we can remove the cell wall? By using cell wall digesting enzymes. By using what? Cell wall digesting enzymes or cell wall hydrolyzing enzymes. This cell wall digesting enzymes or cell wall hydrolyzing enzymes, they digest the cell wall. What gets digested? The cell wall gets digested by using cell wall digesting enzymes or cell wall hydrolyzing enzymes. Is this statement okay? Right? Now listen here. Sir, for example, I am taking a plant cell, sir. Which cell, sir? Plant cell. Plant means prokaryotic or eukaryotic, sir? It's eukaryotic. Plant means it's eukaryotic. It is a eukaryotic. Is it okay? Right. Sir, what is the main component in the cell wall? Main component in what's our cell wall? In plant cell wall, what is the main component? In plant cell wall, the main component is cellulose, right? What is the main component, sir? Cellulose. Therefore, which enzyme we have to use, sir? Cell wall digesting enzyme by name what? Cellulase. Cell wall digesting enzyme. Which cell wall digesting enzyme we have to use here? Cellulase. Cellulase. Now listen the statement. Sir, cellulase acts on what? Cellulose. In first year, we read that this cellulose is a homopolymer of glucose. Yes or no? Cellulose is a homopolymer of glucose. Homopolymer of glucose means made up of many glucose units. Made up of many glucose units. Is it okay? Homopolymer means all monomeric units are same. What are that monomeric units? Glucose. Here how many glucose units are there? Many glucose units. These glucose units, they are linked by which bonds are? Glycosidic bond. These glucose units, they are linked by which bond? Glycosidic bond. Which bonds are? Glycosidic bond. For example, this is cellulose. Cellulose is made up of many glucose units. Cellulose is made up of many glucose units. The bond present between two glucose units is glycosidic bond. When you use cellulase enzyme, that cellulase enzyme breaks this glycosidic bond. Once this glycosidic bond is broken, the glucose units get separated from one another. So cellulose gets hydrolyzed. Is this statement okay? Right. Therefore, enzyme is breaking which bond, sir? Is breaking which bond? Glycosidic bond. Therefore, bond broken. Sir, which bond was broken here, sir? Glycosidic bond. Which bond, sir? Glycosidic bond. Is this statement okay? This is concerned to which cell, sir? Plant cell. Now, if you take a fungal cell. Sir, fungi, prokaryotic or eukaryotic? Sir, it's eukaryotic. It's eukaryotic, right? Right. Sir, in fungi, what is the main component in cell wall, sir? Chitin. The main component in cell wall is what, sir? Chitin. As main component in cell wall is chitin, to hydrolyze the cell wall, we have to use which enzyme, sir? Chitinase. Which enzyme we are using, sir? Chitinase. Chitinase. Sir, this chitinase breaks which bond, sir? Here also it breaks a glycosidic bond only. Which bond it is breaking, sir? Glycosidic bond it breaks. Is it okay? Right. Sir, I am taking a bacterial cell, sir. Which cell, sir? Bacterial cell. Sir, bacteria, is it prokaryotic or eukaryotic, sir? It's prokaryotic, right? It's prokaryotic. Sir, what is the main component, right, in bacterial cell wall? Peptidoglycan. 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 Is it okay? What it is? Peptidoglycan. Sir, peptidoglycan, what is another name for peptidoglycan? It is also called as murin. It is also called as what? Murin. In first year, we read that this peptidoglycan is made up of N-acetyl myramic acid and N-acetyl glucosamine. Yes or no? In first year, we read that this peptidoglycan is made up of N-acetyl glucosamine and N-acetyl myramic acid. What and what, sir? N-acetyl glucosamine and N-acetyl myramic acid. In between N-acetyl glucosamine and N-acetyl myramic acid, which bond is present, sir? Which bond is present? Glycosidic bond is present. Which bond is present, sir? Glycosidic bond is present. Sir, to remove the cell wall, okay, of a bacterial cell, which enzyme we have to use? We have to use lysozyme enzyme. Sir, this lysozyme enzyme breaks which bond, sir? It breaks glycosidic bond that is present between N-acetyl glucosamine and N-acetyl myramic acid of peptidoglycan. Sir, peptidoglycan means made up of N-acetyl glucosamine and N-acetyl myramic acid. In between these two, which bond is present? Glycosidic bond. That bond was broken by whom? Lysozyme enzyme. Lysozyme enzyme. Is this statements okay? Therefore, by using cell wall digesting enzyme or cell wall hydrolyzing enzymes, what gets removed? The cell wall is removed. If it's plant cell, as main component in cell wall is cellulose, the enzyme used for hydrolyzing the cell wall is cellulase. This cellulase breaks glycosidic bond present between glucose units of cellulose. Is it okay? Clear? If cell is fungi, as main component in cell wall is chitin, we are using chitinase enzyme, right? If it's bacterial cell, the cell wall is made up of peptidoglycan, right? Peptidoglycan. So the enzyme used is a lysozyme. What is the enzyme used, sir? Lysozyme. Therefore, by using cell wall digesting enzymes or cell wall hydrolyzing enzymes, the cell wall gets lysed. The cell wall gets lysed. Is it okay? Right? Thank you.